Hello, Krishanjali. Hello. Hello. Can you hear me?
Hello. Yes, sir.
Very nice. So YouTube has been started. You can start.
Okay, sir. Okay, sir. Good morning to everyone. President of Microbial Society, Dr. A.M. Deshmukh, sir, and today's speaker, Dr. Anuja Zine, ma'am, and all other participants. I welcome you on this webinar series. Today is 34th lecture in this PhD webinar series organized by Microbial Society India. And today's speaker is Dr. Anuja Zine, ma'am. Ma'am, currently she is working as assistant professor in Department of Biotechnology, Devgiri College, Aurangabad, and she has completed her PhD in 2020 from Dr. Babasaheb Ambedkar, Marathwada University. Ma'am has published many research papers in reputed journal and also attended various workshops and conferences. Also, ma'am has presented her paper on microbial beta mannanase, future of biofuels at Avishkar in 2016, and also got selected at university level. Ma'am, I welcome you from Microbial Society. And now I request you to start your presentation.
Yes, thank you so much, ma'am. Am I audible?
Yes.
Okay. So the topic of my research is studies on microbial beta mannanase and its application in ethanol production using agro residues. Just a second. So the need and significance of the research. As we all know, India is a country with a great population, and such great population, it requires a great energy demand also. So in India right now, we all know that the vehicles, we can see vehicles everywhere, and all the vehicles, they are using the petrols. But now today also the government are taking lots of measures to control the cost and quantity of the petrol consumption. So the ethanol can be used in the petrol. Right now, today also the government is blending 10% of ethanol in the petrol. So this 10% of ethanol can be increased up to 25% in the future. Means up to 2025, this ethanol blending will be increased from 10 to 25%. So that means what the ethanol which is required in ethanol petrol blending, that is also increasing, the energy demand is also increasing. So there is a need to find more alternative sources for the ethanol production. So we have done a little attempt for the ethanol production using our enzyme. So the beta mannanase is the enzyme which is used for the ethanol production. So beta mannanase. Beta mannanase is the enzyme which is also called as the mannan endo 1,4 beta mannosidase, which is commonly called as the mannanase. It is an endohydrolyzed enzyme, and which is belongs to the member of glycosyl hydrolase families, that is the 5 and 26. This beta mannanase, it mainly acts on the mannan content of the lignocellulosic biomass, which is naturally found in plants, in the trees, in plants. So it this mannanase enzyme, it randomly hydrolyzes this mannan content, and it produces mannooligosaccharides. So this mannooligosaccharides, the main component is the D-mannose. Here you can see the mode of action of that enzyme. It is mainly acting on the beta 1,4 glycosidic linkages. Here you can see, and it is converting this mannan, which is a complex and recalcitrant to degradation, and it is cutting it down and converting into mannooligosaccharides, which main sugar content is the mannose sugar. So there are various applications of this Mannanase enzyme, and it has a large number of industrial applications also, like in detergent industries, in hydrolysis of coffee extract, in paper and pulp industries. In paper and pulp industries, we know that mostly the paper, it is made from the lignocellulosic biomass, like cellulose material is the which is used greatly in this paper industries. So along with the cellulose, lignin and the mannan are also the part of that material, but we need only the cellulose for paper industries. So to degrade and to hydrolyze the mannan contained in the paper industries, that mannanase is utilized. It is also used as a non-nutritional food additive, like prebiotic. It can be utilized, and in poultry feed also, to increase the nutritional value of the poultry feed, this enzyme can be utilized. And obviously now in biofuel production also, this enzyme is taking a great hike. So these are some of the applications of Mannanase enzyme. The study which we have done for the Mannanase producers, for isolation of Mannanase producers, we have screened 50 different soil samples, and the sampling was done in the areas where the lignocellulosic biomass was more, like in farm areas, in garden areas, in cattle shed areas. So we have collected 50 different soil samples, and the enrichment was done for bacteria, fungus, and actinomycetes isolation. The substrate which we have used for the enrichment, that was the 1% copra mill. Copra mill, that means the coconut mill, which was produced in the laboratory itself. And as the coconut is a rich source of mannan, so that's why we have enriched the nutrient broth, potato dextrose broth, and the starch casein broth with this copra mill, so that it will enrich the mannanase producer. The primary and the secondary screening of the isolate was done. In the primary screening, instead of copra mill, now we have used the more standard substrate for the mannanase, that was the guar gum, which was procured from the high media. And this guar gum is a rich source of mannan. So the organisms which were showing the clear zone of hydrolysis around the colonies, they were selected for the secondary screening. So we have primarily screened 50 bacteria, 30 fungus, and 15 actinomycetes, which were the potent producers of the Mannanase enzyme. Depending upon the activity ratio, we have screened primarily the organisms. The organisms which were primarily screened then were subjected to the secondary screening. In the secondary screening, we have used the same media which was used for primary screening, but it was in the liquid form, so the broth form. The guar gum was the sole source of carbon, and another requirement was only the minimal salt medium. The enzyme assay was done using the DNS method, which was given by Miller in 1959. And the optical density of the reaction was taken at 540 nanometers. So these were the three potent isolates which we have got after the screening, primary screening and the secondary screening. And among this, after the secondary screening, we found that the actinomycete isolate AZ12, it was showing much better activity than bacteria and the fungi. And also lot of literature, after the literature review, we have found that much of the research, it was done using the bacteria and the fungal isolate, but much less work was done on actinomycete. Actinomycetes. So we have done what? We have selected the actinomycetes for our further research, because less area was explored, and it was a great opportunity for us to explore the actinomycetes in this enzyme. So we have selected the actinomycete AZ12. Then we have identified that isolate, that is AZ12. Morphological identification was done. Then biochemical identification was done, and genotypic analysis was done for the complete identification of that organism. So this is the morphological examination of the potent isolate. Yeah, these are the colony characteristics. When we were doing the morphological examination, we found that this actinomycete was able to produce the diffusible pigment, which was reddish brown in color. And this is a microscopic examination of the actinomycetes, where the mycelial growth was somewhat curve shaped. Then the biochemical characterization of the actinomycetes was also done, in which its temperature tolerance, pH tolerance, enzymatic studies was also done. So in temperature tolerance, we found that the actinomycete showed the much better growth at 40 degrees Celsius, and its optimum pH was 8 which was slightly alkaline. And in enzymatic studies, we have studied different enzymes like amylase, urease, catalase, oxidase, and so on. The IMVIC test was also performed. Then we have also studied the GC content of that Actinomycete, which was found to be 60.48, and its melting temperature was 90 degrees Celsius. For the genotypic analysis, based on the nucleotide homology and phylogenetic analysis, the selected Actinomycete was identified as Streptomyces flavius strain, and the sequence of this submitted in the gene bank with accession number of K5212951. So this was a phylogenetic tree which was done with the help of blast results from NCBI. Then again, now we have used the standard substrate, that was the guar gum. Now to again reduce the cost of the Mannanase enzyme, we have used different agro residues as a substrate and as a source of mannan. So we have screened 25 different agro residues, out of which we have studied 50 different agro residues, but out of which 25 were selected, which were rich source of mannan. So we have used the production media. Again, it was a minimal salt media with 1% of agro residues. These agro residues were shed dried and they were powdered into fine powder, and it was used as a source of mannan. So from the results we got that the soybean straw, it showed the much enzyme activity than other agro residues. So we have used the soybean straw as the enzyme substrate for our further productions, reduce the cost. Then before going for the production process, we have done the comparative study of solid state and submerged state fermentation. So whether the enzyme activity is much in solid state or submerged state, that we have studied, and we found out that the enzyme activity, the enzyme required more aeration for its production. So the submerged state fermentation was selected for a further enzyme production process. Then optimization of the process parameters for enhanced Mannanase production using the potent isolate, that is Streptomyces species AZ12, was done. The process parameters like the physical parameters and the nutritional parameters were studied. In physical parameters, the different media, inoculum, age, size, period, pH, and temperature were done. And for the nutritional parameters, for optimization of nutritional parameters, the agro residues, to in how much percentage it should be used, that was studied. Organic, inorganic, nitrogen sources required for production, then inducers, NaCl concentrations, all these were studied. So in media optimization, we have studied different types of the media. 6 different types of media were taken, and from that we found out that medium number 6 showed the maximum enzyme activity than other medium. So we have selected the medium number 6 as the optimum media for the enzyme production. Then the process parameters like inoculum age, inoculum volume, incubation period, temperature, pH, it was studied. And these are the results for that. Then after the process parameters, now it was a main thing to optimize the media components. Means we have selected the media, but how much quantity the media components should be used for the maximum production of the enzyme, it was done. So we have used some statistical design for optimizing the media components. So by using the statistical design, we have reduced the process variability, and even we have reduced the development time of that enzyme, and we have also improved the product yields. So the statistical design has helped us a lot in optimizing the media. For the statistical designs, we have used 2 softwares, like Plackett-Burman design was used and the central composite design was used. The Plackett-Burman design, it was done using Minitab software. And for central composite design, we have used the Design Expert software. So by doing the Plackett-Burman, we will get to know the importance of various factors. Means for enzyme production, which factors are very significant, that we will come to know by using the Plackett-Burman design. But to get the exact optimum value of that factors, the central composite design helps a lot. So both the designs, statistical designs are very important for the media optimization. So this is the design for the Plackett-Berman in Minitab software. We have adjusted the low level and the high level values of all the components. Here we can see the components. So here now we have used two source of carbons, that is soya peels were also used as a carbon source because that was optimum in our agro residues, and copra meal was also used because we know that both these sources are rich in mannan. Then the nitrogen sources were asparagine, soya seeds, and the KNO3, and other were the trace elements which are required for the growth of organism. So likewise, this Plackett-Berman design was designed for the optimization. Then this is the design which was given by the Minitab software. 12 different runs were given by the software. And after running this 12 runs in different flasks, 12 different flasks, the production was carried out. And we came to know that in flask number 6, 7, 9, and 12, the production was much better than the predicted response which was given by the software. So these are the results of the Plackett-Berman design. Then the p-value. Now, whatever the statistical optimization is there, it is mainly done by the p-value. Means if the p-value is 0.05 or less than 0.05, those factors are considered as the significant factors. So from this Pareto chart, which was given by the software, we can see that the soya seeds and the soya straw were found out to be the significant factors which are responsible for the Mannanase production. So from this, we came to know that the soya straw and soya seeds have a great effect on the production. Then after getting the significant factors, we should know the optimum value of that factors. For that, the CCD or response surface methodology was used. And the software which we have used for this design, that was the design expert software. So the software has given us 13 different runs, from which the flask number 8, it showed the maximum enzyme activity. So this is the ANOVA response also, which was given by the software, and it showed that the model which we have designed for optimization, it was the significant. So this is the response surface plot, which is given by the software. It shows the cumulative effect of our factors, and it shows the response, means how the factors are effective. It gives a 3-dimensional surface plot. Again, the software has also given some predicted enzyme units. It has given the point prediction, and in which they have given us the values of soya straw and soya seeds. So we have run the flask kit, the triplicates, and we found that the enzyme units which we have got are very resembling to the predicted enzyme units. And this shows that the experiment validity is 95.88%, which is much better. So we have again studied the optimized and the unoptimized media. The optimized media, after the Plackett-Berman design and central composite design, the enzyme units we found that were 163.9 units per ml, and in unoptimized it was only 55.82. That means there is a 3-fold increase in the enzyme activity with the optimized media as compared to the unoptimized media. So the optimization was done, and then we have subjected our strain for the strain improvement process. So in the strain improvement, we have used many certain chemical mutagens and physical mutagens. In chemical mutagens, we have tried with ethidium bromide, sodium azide, and as a physical mutagen, we have used UV irradiations. But the UV radiations and the ethidium bromide, they have not showed much effect on our enzyme activity, and there were not much variations in the results. So we have not selected these procedures for further mutation purpose. But with the help of the sodium azide, the colonies had showed a change in morphology above the LD50 dose, and we have selected that colonies for enzyme production. So from that colonies, AZM8 colony showed improved enzyme production than the wild type. So we have selected that AZM8 for our production process. Means we have studied a few generations, we have studied with this AZM8 and saw the stability of production. And then we have selected this AZMate for our further studies of the enzyme. So both wild type and mutant strain was used for the production, but the mutant showed much more production than the wild type. Then purification of the enzyme was done. Purification was done with the help of different techniques like ammonium sulfate precipitation, partially purified, the dialysis method, ion exchange chromatography, and gel filtration chromatography were used. And the purification fold was reached up to 7.67. Then this is the methods which we have used for the study of enzyme activity, that is the DNS method. And for protein estimation, we have used the Follin-Laurie method. This is the graphical representation of the gel filtration chromatography as well as the ion exchange chromatography. Then after the purification of the samples from the gel filtration chromatography, we have determined the molecular mass of that beta mannanase, and it was found that the molecular weight of the enzyme was 42 kilodaltons. Okay. The SDS page was performed. Some Zymogram studies was also done where this enzyme substrate reaction can be seen clearly after staining with Congo red solution. Then after the purification process, we have characterized that enzyme for the effect of temperature, pH, reaction time, substrate concentration, and metal ions. So the characterization of the enzyme was done, in which we have compared both wild type as well as the mutant strain. So in case of the mutant strain, the enzyme activity was much more than the wild type, and the optimum temperature for the enzyme substrate reaction, that was the 50 degrees Celsius, and the pH stability was also checked. Then effect of pH was also checked, and the optimum pH was found to be 8, which is slightly alkaline with the mutant strain, and optimum pH with the wild type, it was 7. Then pH stability studies were also done. The reaction time, the enzyme substrate reaction time, it was 60 minutes in case of the wild type, but in case of the mutant, it has reduced with 10 minutes, that is up to 50 minutes. Then the optimization of the substrate concentration was done. Here we can see the optimization of the substrate concentration, and the Km Vmax was also determined using that graph. Then the effect of metal ions was also studied, and its relative activity was also studied, and it was found that the Fe3 plus ions has much effect or induce the enzyme activity. Then this was the Km and Vmax, which was calculated for both the enzymes, wild type as well as the improved. So the decrease in Km for the improved strain, it indicated that more enzyme substrate affinity as compared to the wild type, and thus there is also increase in the Vmax as compared to the wild type. So the strain improvement has improved the enzyme efficiency. We can conclude that the strain improvement was done properly. Then after the optimization processes, we have also studied the immobilization of the enzyme. For the immobilization purpose, we have used 3 different carriers. Sodium alginate was used, gelatin was also tried to use, and chitosan was used. But in case of sodium alginate, we didn't got much enzyme activity, so it was not selected for the immobilization purpose. Gelatin, it did not sustain at such a high temperature, that is 50 degrees Celsius. That's why the gelatin was rejected during the experiments. But the chitosan, it sustained the temperature also, and it has given the highest enzyme activity also. This chitosan bits use the covalent linkages for the to bind with the matrix, and we have used the chitosan as an immobilizing agent. Then we have checked the stability of the enzyme, immobilized enzyme, is for how many days this enzyme remains active in immobilized state. So we can reuse this enzyme even after 2 months also. We can see that for every 2 months we have checked the enzyme activity, and it was near about same. Then later on, after 3 months, after 4 months, the enzyme activity started decreasing. Then the scale-up program was also done. from 10 ml to 1500 ml flask. And during the scale up also, we found that the enzyme was stable in all the flasks. The enzyme activity was stable. The percent activity was stable in all the flasks. Then finally, after the production process, the Mannanase enzyme, it was applied for the ethanol production. So what we have done, we have used the two methods for the production process. One is the pre-saccharification process, and the another is the simultaneous saccharification and fermentation process. So this process was also optimized statistically to get the maximum yield of ethanol. And the purification of the ethanol, we have used the distillation process, which is the most dominant process at the industrial level also. And we have done the statistical optimization for the ethanol production. Before that, we have comparatively studied pre-saccharification and simultaneous saccharification and fermentation process. In case of pre-saccharification, what we have done, we have first added the enzyme units in the flask containing the substrate. And we have first of all prepared the mono oligosaccharides, and those mono oligosaccharides were then subjected to ethanol production using the Saccharomyces cerevisiae in pre-saccharification. But in case of simultaneous saccharification and fermentation process, we have added the enzyme units along with the Saccharomyces cerevisiae inoculum, so that the simultaneous saccharification will be there using the enzyme units, and as the mono oligosaccharides are produced, the yeast will convert it into the ethanol. So that was the motto of that experiment. So by a comparative study of this, we found that the simultaneous saccharification and fermentation process was gave the much better results or ethanol yield as compared to pre-saccharification process. So we have used our ethanol production studies using the simultaneous saccharification and fermentation process. So again, the Plackett-Berman design was designed for optimization of the process parameters for ethanol production, and the enzyme concentration, substrate concentration, inoculum concentration of the yeast, temperature, incubation time, these were the variables which were used for the ethanol production. So the low level and the high level was set, and the software has given us 8 different runs, out of which the flask 2, 4, 5, and 8, these runs shows more ethanol production than the predicted response which was given by the software. So here you can see the results. Again, the significant factors using the Pareto chart, the factors which are showing the effect, p-value less than 0.05, those were considered as the significant factors. So here the substrate concentration and the enzyme concentration, they were found to be significant for the ethanol production. So from the Pareto chart, we come to know about the significant factors. Then that significant factors to calculate the exact values which should be used for the production, that has been studied using the CCD design. So design expert again was used for the design expert was again used for the CCD design. Here 13 different runs were given for the by the software, and 13 different flasks with the respective optimum conditions were inoculated and placed, and the ethanol production was checked. Again, with the help of the analysis of variance, we found that the model which we are studying for the ethanol production, that was significant. Then this is against the response surface plot of the ethanol, which is showing the cumulative effect of substrate concentration and the enzyme concentration to give the highest ethanol production. Then we have also done the validation of the optimized condition, where we found that the ethanol production after the optimized condition, it was 12.26, which is quite close to the predicted value which was given by the software. Hence, the model which we have chosen for the optimization, it was right correct. Then after the optimization process, we got the exact values, how much substrate concentration should be used, how much the inoculum should be used, all things were set, and then we went for the production of the ethanol. So during the production of ethanol, we have used that simultaneous saccharification and fermentation process. And the standard culture of Saccharomyces cerevisiae was used for the production of ethanol. So we have added the enzyme, and we have enzyme in units which were optimized, and we have also added simultaneously the Saccharomyces cerevisiae into the production flask using the soya straw as a substrate, and the alcohol production was done in anaerobic conditions. So after the production, we have distilled the ethanol using the distillation unit in the laboratory, and the dichromat test was done, triiodomethane test was done for the confirmation of the ethanol. Then specific gravity method was used for calculating the percentage of alcohol from that agro residue, that is a soya straw, and it was found to be 19.26%, which was much better than the other results, other literature reviews which we have read so far. So 19.26% of the ethanol was used. The HPLC was also done of the filter of our filtrate distilled sample. So these are the results of HPLC. Then the GCMS was also done of the fermented distillate. So these are the results of the GCMS analysis. And the production of the ethanol, it was done successfully using the optimized conditions and the enzyme, that is the beta mannanase. So in this way, the whole production of the ethanol using our enzyme which we have tried, it was successful. And some future perspectives are there, we have to do much more work on this field, that is the production of mannanase on a large scale. We are planning for that. The whole genome analysis of Streptomyces flavius AZ12, which was main source of the mannanase, and also more efficiently on a large scale ethanol production, and how again we can increase the yield of the ethanol. On that we have to study much more. And yes, of course, the application of ethanol as a biofuel, if we can do that, we are going to do that also. So thank you so much everyone for giving me the opportunity to use such a big platform, that is the Microbial Society of India. If you have any questions, you are free to ask. Thank you so much.
Okay, thank you, ma'am,
Yes.
for giving presentation. Now we'll move towards question answers. Participants, if you have any question, you may ask directly.
Hello. Hello.
Yes, sir.
I'm Dr. Swaranjit here from Chandigarh.
Yes, sir. Yes, sir.
It was a very good talk.
Thank you so much,
really
sir.
Enjoyed every bit of it.
Sir.
I really enjoyed. Your
Thank
name
you,
is
sir.
Anuja, right?
Yes, sir.
Okay. Which was the best substrate you used? The best one?
Best substrate. Standard substrate you're talking about, or agro residues in terms of
the growth substrate.
Okay,
for
growth substrate.
Yeah.
So for growth substrate, we have used the mannan itself. Means the mannan was the main source. If the organism is utilizing mannan, then we can go for mannanase production. So for the as a best substrate, till now we have used the guar gum.
Okay.
That was the only rich in mannan, and it showed much activity also, and the growth was also much better on the mannan containing media. So we have used the guar gum.
What was the coconut substrate you used? Was it good, the coconut one?
Yes, the copra mill was also good, but the thing was that the residues of the copra mill, they were interfering in somewhat in the growth. So that's why we skipped the copra mill.
Okay.
And we have turned towards the guar gum.
I see. Very good. Is your enzyme thermostable?
Yes, sir. It is means quite thermostable. Means in all temperature also it is showing much good activity.
At what temperature?
Mostly the optimum temperature for our enzyme was, sir, 40 degrees Celsius.
Good, good.
At 40 degrees Celsius, it was showing much higher activity. Even at 50, 60 degrees Celsius also the enzyme activity is not lost much. It is showing activity up to 80 degrees Celsius also. Means it is like,
So
you can
that
say
means
thermotolerant. Yeah.
that means it can be used in thermo conditions
Yes.
also.
Yes. Yes. Yes.
Okay. Can it bear low temperature like 20 degree or 15 degree?
No, actually at 10, we have started our enzyme activity. Means we have studied that also. Here you can see in the graph the temperature from 10 degrees Celsius, 20 degrees Celsius, but the enzyme activity was not much more, sir, in lower
Okay.
temperatures.
Okay.
So
And
that's why we have used.
this is with bacteria or actinomycetes?
No, all the work which I have done, that is about the actinomycetes, sir.
Okay, no bacteria.
No, sir. We have in initial phase we have studied only which bacteria were potent. If it would have shown much more results than actinomycetes, then we would have selected bacteria. But actinomycetes showed much better results than bacteria. That's why we have selected actinomycetes.
Does your strain have a plasmid?
So the no, sir. We have not studied that. We have just extracted the DNA and we have gone for the genotypic analysis.
No, what I'm saying, if your strain is having a plasmid, you can engineer it for growth at low temperature,
Yes.
which is very useful in psychrophilic conditions like Leh and Ladakh and Western countries which are having very low temperature, you know.
Yes.
So
Yes.
your enzyme can also be made at psychrophilic condition.
Yes, sir.
So
Yes, sir.
think
We
about
will
it.
definitely. Yes, sir.
You think about it
Definitely, sir.
later on.
In future perspective, I will keep that point in mind, sir.
Yeah, this is very important and you will get a lot of value for your work.
Yes. Thank you so much, sir.
Thank you. Thank you.
Yes. Yes. Thank you, sir.
Anuja, can you hear?
Yes, sir.
Very wonderful work starting with the isolating the potent organism characterization and especially the characterizing the enzyme and
Yes.
looking for a mutant. And
Yes.
you have characterized the mutant also and brought it into the practice, especially with reference to the ethanol production.
Yes,
This is
sir.
definitely has a lot of commercial application, right?
Yes, sir. Yes, sir.
Just I wanted to inform that beta mannanase has been used for prebiotic activity also. No,
Yes, sir.
galactomannan production.
Yes, sir.
Galactomannan has found to be a very potent prebiotic molecule commercially used these days.
Yes,
Actually,
sir.
my question was not related to that. Just how you actually selected the mutant, because I don't see a much better activity compared to the wild type. So, what was the basis of initial screening the mutant?
Initial screening of
If you have
mutant.
the slide, also will be much easier to understand, perhaps.
Yes, sir. Yes, sir. Just a second. Yes, sir. This. Sir, actually, the thing which we have done after the strain improvement, the organism
That
which
the
we have,
sodium azide induced mutant. In
yes,
fact, I put this
it's
question in a chat box also. If you
okay.
can just show me your mutant studies, it will be very convincing. Or maybe if somebody wants to isolate the mutant, perhaps they have to go for a rigorous screening. Means screening the more numbers, looking for the potent one, seeing its growth characteristics, and like that way.
Okay. So for selection of mutants,
And
sir,
then
we have,
from this sodium azide induced one, what was the lethal dose you gave?
sir, it was 62 ppm. Lethal dose was
Percent
62 ppm.
mortality. Oh, wonderful. Yes.
Yes.
Yeah, looks quite all right. Exactly.
Yes, sir.
Nearly 50 was found to be, yeah, because I expected that you should have given the dose of somewhere 90, 99%, only 1 or 2% survival are there. And then from there, if you check, perhaps you will
Yes.
get the better mutant, which will have improved function, improved enzyme activity.
Improved enzyme activity also. Yes, sir.
And then you have the enzyme production of the wild type versus mutant, right?
Yes,
In
we
the
have studied that.
So percent survival is there. and then mutant, exactly.
Yes,
Yes,
sir.
yes.
So on the basis of this
Yeah,
only,
wonderful.
sir, we have studied the mutant organisms, sir.
You use the Gavargam only for all the studies till the ethanol production also?
No, sir, we have altered the substrate, sir. I
Okay,
told you
wonderful.
that we have used agro residue for ethanol production.
Oh,
We
yeah,
have
yeah.
studied the agro residues, 25 different agro residues were screened, and then we have selected the soya straw for the
Okay.
production of ethanol also, sir.
Mm-hmm.
Yes.
Wonderful, very good study.
Yes, thank you so much, sir.
I think there is one more question on the chat box. If you can
Okay.
read.
Yes, yes. I'll jump to first unread message. Okay.
From Nilima.
From Nilima. Great work. Nilima, yes, it was. Did you characterize the enzyme with respect to type of Mannanase? No, ma'am. Nilima, ma'am. We have not characterized with respect to Mannanase, but we have characterized what enzyme our organism has produced. That we have done. With respect to other Mannanase which were produced by other bacteria, we have not compared that. So, thank you so much for the question. Any more question if you have? So I think there are no more questions, right? So we can conclude.
Hello, hello, ma'am.
Yes, ma'am.
Okay. I think there is no question from participant side.
Yes.
Mm, mm. Mm. Should we wait for 1 to 2 minutes or should we continue? Should we stop?
We can conclude, ma'am. No problem.
There is one more question
Okay.
from Nilima ma'am.
Yes.
How much is the substrate to the product conversion rate with this manganese in alcohol in?
Just how much is the substrate to product conversion rate with this Mannanase? Actually, there is more than 60% of the substrate, more than 60 to 70% of the substrate is converted during the alcohol production. And we have used the standard enzyme for the conversion of this substrate into alcohol. So that's why the rate was much higher according to our studies, ma'am, Nilima ma'am. And that's why only the alcohol production was much better, because the enzyme, it acted on the substrate and then it produced the mano oligosaccharides, and that mano oligosaccharides were converted into ethanol. Means it was such a long process, but still our enzyme worked very efficiently, and it has degraded or hydrolyzed that substrate, and we have used or we have got the yield of alcohol much better, ma'am, as compared to other substrates. With crude extract, you can also get, but there are more interfering factors in the crude extract also. But you can go for the ethanol production by using the crude extract also, ma'am. So we can get much better, because in the crude extract, it is not the purified enzyme. You can get some factors of other enzymes also, which can also help the production and which can also help to act on the substrate. So that might be, means you can also use the crude extract more efficiently. We can use it. But we want to show the effect of our enzyme itself. That's why we have used the purified enzyme. But we can use the crude extract also for our production process, ma'am. Which design, ma'am? Which design you are talking about? Means the placket Berman design, which we have experimental design. Okay, means we have used statistical design for the optimization purpose. The reason was that to get the more optimum conditions, and because it is very less time consuming. One at a time approach takes lot of time, more days will be involved in that. That's why we have used the you can say the statistical softwares for the optimization purpose. But by one at a time approach also you can get the exact optimum conditions. That is the very traditional method we use in the laboratories also nowadays. But for research, the software is used. The software is freely available. Yes, it is freely available on the internet also, but now the software are taking like we have to pay some money to get access to that software. But some softwares are there that are freely available also, ma'am, on the internet. Yes, thank you. Thank you so much, ma'am.
Anuja, just a small more, one more
Yes,
addition. My mic was actually off.
yes, sir. Yes, sir.
You are testing the prebiotic activity of this Mannanase, because
Okay,
I have
sir.
very good setup a probiotic bacteria. So actually, I can
Okay.
actually, so you have suppose a Mannan treated with your Mannanase, and then you get a beta galactomannan, right? So
Yes,
that
sir. Yes, sir.
can be checked exclusively by the lactic acid bacteria and then bifidobacteria, and then we can their hydrolysis as well as short chain fatty acid production. So if you are if you are extending this study further, let us know.
Yes. Yes, sir. Definitely, sir. We will enjoy working together, sir. Definitely, I'll contact you, sir.
Yes.
Thank you so much. Yes, sir.
Hello, anyone having question? Participant, do you have any question? You may ask. Okay, ma'am. I think there is no question.
Yes.
So now first of all, I thank you, ma'am, for giving your time, and it was a great presentation. Your work is also very good, and also I wish best luck for your future. And also I thank to all other participants for being with us. Thank you all.
Yes, thank you so much for giving me the opportunity to present my work on such a great platform. And I really want to thank Dr. Arvind Deshmukh, sir, the president of Microbial Society of India, for giving me such an opportunity. Thank you so much.
Thank you.